So characteristics of connective tissue. Connective tissue is kind of weird because it's a whole host of various types of cells and tissues that get lump summed together. In general, connective tissue is going to be made up of a looser extracellular matrix, where this extracellular matrix can be solid, semi-solid, or liquid in nature, but it always has some sort of protein within a very large amount of extracellular matrix. The cells that are going to be making connective tissue we refer to as being fibroblasts or fibroblast-derived cells. And so what we're saying fibroblasts is that these are cells that are going to build fibers. Now, even though we call them fibroblasts, there's a whole host of subsets of cells within that, and we'll talk about those subsets when we actually look at the distinct systems that are going to be derived of these types of connective tissue. So when we look at connective tissue itself, it's going to get broken down into three distinct classifications. All connective tissue is going to derive from what's referred to as the mesenchyme or mesenchymal cells. The connective tissue is to be broken down into what's referred to as connective tissue proper. These are tissues that actually will connect one thing to another thing. Then we have what's referred to as supporting connective tissue. These are connective tissues that build the structures of the body. In particular, the skeletal structures and the cartilaginous structures of the body. And then we finally have what's referred to as the fluid connective tissue. And the fluid connective tissue is going to be connective tissue that allows for communication and movement of materials through the body. So when we look at connective tissue proper, we break it up into two distinct classifications. The two distinct classifications we have what's referred to as the loose connective tissue. In the loose connective tissue, what we have is we have fewer fibers and more what's referred to as ground substance. That is the fluid space within the matrix itself of the proteins. This includes what's referred to as areolar connective tissue and reticular connective tissue. And then we toss in another type of tissue that you won't think of being a connective tissue, even though it's classified as being connective tissue, and that is adipose tissue. And adipose tissue doesn't have a lot of actual proteins that surround the adipose cells themselves, and so that's why we stick it in with the loose connective tissue. The other is going to be the dense connective tissue, and the dense connective tissue tends to have more protein fibers in the extracellular matrix and less fluid within the extracellular matrix. This is going to include things like what's referred to as regular dense connective tissue and irregular dense connective tissue. And then we have what's referred to as elastic connective tissue. So all of these connective tissues here, the connective tissue proper, is going to be made up of three principal proteins. It's going to be made up of collagen, elastin, and reticular protein. And the combination of those proteins is going to dictate the characteristics of the connective tissue that we see. Leaving the connective tissue proper, let's go ahead and look at the supporting connective tissue. In this case here, we break it up into two parts, and that is how solid is this actual extracellular matrix? That is, does it give us a rigid structure or does it give us a semi-rigid structure? In the semi-rigid structure, we have what's referred to as cartilage. Cartilage is an avascular tissue, which means that its ability to do metabolism and to go about doing repair of the cell itself is based off the ability to diffuse things into the lacunae where the cartilaginous cell happens to be, the chondrocyte. Because of that, cartilage is going to be less apt to repair itself than bone. And we'll talk more about this when we look at the actual skeletal structures and bone tissue when we look at the musculoskeletal stuff. When we look at cartilage, we have three distinct types, and the three distinct types are the hyaline, the fibro, and the elastic. And the difference between the hyaline, the fibro, and the elastic is how the fibers are arranged, and then what types of fibers we're going to see, what types of proteins we're going to see. In the elastic, we see a lot more elastin protein, whereas in the hyaline and the fibro, we see less elastin. The other type is the bone, and the bone is a very solid extracellular matrix. It's predominantly going to be collagen proteins that are held in place by a crystalline structure known as hydroxyapatite. And the hydroxyapatite arranges itself in such a way so that it is able to uh, withstand shear, torsional, compression, and tensional forces placed on the bone itself. There are two distinct types. Unfortunately, books give weird names to these, and so let's use the correct terminology here. We have what's referred to as cortical, and the cortical bone is the bone that's in the outer ringed area of the bone. And then we have trabecular, and trabecular is little kind of spikes that we see onto the internal side of the bone or onto the medial side of the bone. Books will reference cortical as being compact and trabecular as being spongy. There is no compact bone, there is no spongy bone. All bone is compact, and we'll get more detail into that when we actually look at the bone tissue itself. So what you want to do is go ahead and correct and reclassify the bone types as being cortical and trabecular, Trabecular. The last type of connective tissue was referred to as the fluid connective tissue. And so the fluid connective tissue is going to be the connective tissue that's going to move materials around the body. The predominant fluid connective tissue is blood. However, lymph will also fall underneath this classification of fluid connective tissue. Within the fluid connective tissue, we will have distinct types of cells. We have erythrocytes, leukocytes, thrombocytes. Within the blood, thrombocytes or megakaryocytes are also referred to as platelets. In the lymph, we have lymphocytes. Now, lymphocytes can also be classified within the leukocytes. Now, we have to be very careful here in terms of our designation, in terms of correct terminology again. We do not have red blood cells. We do not have white blood cells. We have blood cells. We reference erythrocytes when talking with our patients as red blood cells. However, we do not, in this class, reference red blood cells. We reference them as being erythrocytes, and if you want to use an abbreviation, capital R, capital B, capital C. And we have leukocytes. Leukocytes is what you'll talk about with your patients as being white blood cells. They're not white, and that's a big, huge issue here. And so one of the things that we'll talk about when we look at blood is that there's no real clear delineation in terms of coloration of white and red. There's a smattering of different colors within that. You can use the abbreviation WBC when referencing the leukocytes. So let's take a quick look at what these cells what this tissue would look like underneath the slide, as slides, and then we'll get into a little bit more detail when we look at the slides themselves in lab setting. So we have the dense connective tissue, and what you notice with the dense connective tissue with both the elastic and the dense regular connective tissue is this regular pattern of arrangement. And so within the connective tissue, we will have a whole bunch of collagen proteins. In elastic connective tissue, like what we see here with the aorta, you'll notice that we have this elastin fiber interwoven with the collagen fibers, giving kind of this purplish, pinkish hue to the slide. Most elastin protein will dye in a darker purple color, whereas most collagen will dye in kind of a reddish, pinkish, purplish color. In the dense regular, notice that we have these kind of waved lines. Now notice all these little kind of circles that we see in the slides. Those are all of the fibroblasts themselves. And so one of the things that becomes kind of tricky is differentiating, say, a dense regular connective tissue from, say, skeletal or cardiac muscle, because they look very similar on first examination. What you have to do is you have to reference where you see the fibroblasts and then how the banding pattern happens to come about. Now notice the difference between the dense regular connective tissue and the dense irregular connective tissue. The difference that we see here is the banding pattern that we have within the tissue itself. Dense regular connective tissue is there to hold things together without allowing a large amount of motion between things. So we see dense regular connective tissue in things like ligaments and tendons. Whereas dense irregular allows for compression to take place in and around the tissues that are being held together. And so we see this in things like the dermis of the integument in the skin. So that's the dense connective tissue. Let's take a look at the loose connective tissue. And so with loose connective tissue, we have our areolar connective tissue, sometimes referred to as loose areolar connective tissue. And so notice what we have is we have these smattering of fibroblasts with long tangential fibers heading away from them, a combination of collagen and elastin, but we have these spaces. The spaces within the areolar connective tissue allows for structures to pass through the tissue. And so blood vessels and nerves will be passing along through the loose areolar connective tissue. This loose areolar connective tissue is what's going to be the underbinding of the hypodermis, and it's going to be attaching the skin to, say, the underlying muscle. If you've ever skinned chicken or skinned beef or pork when you're cooking, and that kind of spiderwebby material comes away from the skin or comes away from the muscle itself, that is loose areolar connective tissue. Now, reticular connective tissue have these very dark, thick reticular proteins within them. And what they do is that they form a rigid structure within this extracellular matrix of space. We see these reticular fibers in areas where we want fluid to move, but we don't want the organ itself to swell. And so we'll see these in lymph vessels, in lymph nodes, and we'll see these in the spleen, where it's going to direct flow into distinct areas that will allow for swelling to take place, but will not allow the entirety of the organ to swell. Now, look up at the top and we see the adipose tissue. And so when the adipose tissue, we have cell membrane to cell membrane contact. We don't have a whole bunch of space for proteins to take up within the extracellular matrix, which is why we stick it with the loose connective tissue. Most of what we see appears to be empty space, but it's not really empty space. What it is a microbody, a vacuole, that has large lipid droplets contained within it. And as the large lipid droplet fills more of the space of the cell, it pushes all the rest of the organelles peripherally, including the nucleus. And so you notice the adipocyte nucleus stuck to the periphery. The last are the structurals. And so let's take a look at the three distinct types of cartilage: the hyaline, the elastin, and the fibrocartilage. And so notice the differential arrangements of the lacunae, the circles within this semi-solid matrix. And what the lacunae is, it's an area for the cell to lie dormant until it's metabolically initiated in order to do repair or grow within this solid mass. In the center of the lacunae, we have the cell responsible for the cartilage, the chondrocytes. Notice the arrangement of the chondrocyte and lacunae and the alignment that each have within the areas of the matrix. Notice how that's different from the bone. In the bone, what we have is we have what's referred to as a concentrically ringed lamina. This concentrically ringed lamina forms what's referred to as the Hervatian system. In each one of those little dots on the slide is an osteocyte. The osteocytes are connected together by what's referred to as the canicules, and the canicules are connected to a canal system. The Hervatian canal, or the central canal, within the center of the concentric ring lamina, and then the Volkmann's, or the perforating canal, which connects to the center medullary region of the bone. This allows for blood flow to get all the way into the canalicles. This means that the bone cells are able to obtain and exchange materials quite easily and not be reliant upon diffusion of materials through the matrix the way in which cartilage is. This is why bone will heal at a much faster rate than cartilage will. And we'll get more detail into this when we actually look at the function of the skeleton. So the last one we have to look at is blood. Blood is the principal fluid or liquid connective tissue in which we have distinct cells. We have erythrocytes, and then we have the leukocytes, and we have four predominant leukocytes. These are the basophils, eosinophils, neutrophils, monocytes. And then we have the thrombocytes, also known as the megakaryocytes or the platelets. And then finally we have the lymphocytes. And so when we look at blood, we usually look at it in terms of a smear. And on this, we have all of the distinct types of cells, where the predominant cell that we see, the cell that we see the most of, are the erythrocytes. And then we see the cells that have these kind of purple horseshoes in them. Those are the neutrophils. And then we see the ones that have kind of like a purplish speckled area surrounded by a pinkish speckled area. Those are the eosinophils. And then we have the basophils, which are the kind of the circled purplish regions. And then we finally have the monocyte, which is the largest of those cells with the single large vacuole in the center of it. And then we see all these little purple and pink dots spaced throughout. Those are the platelets. And so once again, we'll walk through each one of these histological slides in more detail when we get into lab. And as we walk through them in lab, we'll go into a little bit more detail as to what each one will functionally be able to do for the body.